Well, if you want to define nanoscience, I think it really depends on who you ask. I think of nanoscience as the synthesis and the manipulation of very small objects. It's got to be reproducible, and these nanoparticles should have predictable behavior, predictable properties. Another way you could look at it, or another, would be bringing a new perspective to some very well-established fields. As an example, we understand very well what DNA looks like, what it's for, how it functions, and of course there's new things all the time. But now DNA can be thought of as a structural nanomaterial. In other words, it can be used as a building block to in the construction of other materials. So we go from the substance we know, DNA, and now we start using it in a different way. So DNA can be used as a structural unit in larger structures. So you can think of it as just like a spacer with defined size, defined shape, and very well-defined chemical structure. And so it's being used in various applications. There are some sensor applications out there, but I wouldn't want to get too far into it. It's not my area. If you want to think about nanoscience, you got to think about very, very small things. All right. So everybody will always say, well, 1 nanometer is 1/1000th of a meter, but it helps if you can put that in perspective. Okay. So think of a hair, a human hair. Right? You can see human hair, but it's very fine, less than 1 millimeter in diameter. Really, it's about 1/10 of a millimeter. That human hair is approximately then 100,000 nanometers. Okay, well, in diameter. That may not mean anything yet. So the objects that I work with in my research, the particles I work with, are about 1,000 times smaller than the width of that human hair. So what that means is you take the materials I work with and line them up like this, line them up side to side. There would be 1,000 of them across, and that would make up the width of 1 human hair. First, I got involved in the field of conducting polymers. And that was through the postdoc work I did with Dr. Alan McDiarmid at UPenn. He shared the Nobel Prize in 2000 for chemistry, so he's like the sort of one of the founding people in the field of conducting polymers. So I had an opportunity to work with him, and he was actually very interested in some new work that was being done where conducting polymers were being looked at from a nanoscale perspective. So that was how I got my first taste of it. Doing that work, I actually found I had fun playing with the toys that we get to use to be able to image the different kinds of nanoparticles. Of course, you can't see them with your eyes. At best, you can see very large aggregates of lots and lots and lots of nanoparticles. So you're never going to have in your hand one single nanoparticle. It's going to be a huge cluster of these things jammed together. But we have great microscopes, electron microscopes, that allow you to see these individual particles. If you want to think bigger picture, you can say, well, why is it important to be able to do this, to look at these kinds of particles, or to be able to really to make these kind of particles? And here's what I think. The smallest particles that we can now reliably synthesize are on the same size scale as the largest biological molecules. Right? Same size. So that means that you now have a way to directly interact with biological molecules, right? Something synthetic that can interact directly with something natural. So this gives you ways to grab things and work with them. So we could, if you think applications, we could better understand different biological processes. We can interact with these biological processes. So if you think about photosynthesis, the most effective way of taking sunlight and converting it into energy, but it's difficult to reproduce. It hasn't been done. But maybe you have some nanoparticle that can interface directly with that biological system and just take that energy, take the energy that's available and use it. There's the potential to improve detection of various diseases and really ultimately heal the body with really, really, really great precision. With applications, you sometimes have to be careful because nano is a buzzword, and you'll see it everywhere. I've seen it, on bottles of spray cleaners now with, I don't know, nano cleaning foam. And so you always have to wonder, well, is that really a legitimate nano application? But there are things out there. There's, I've seen, you've probably seen also commercials on television for washing machines that use nano silver technology for its antibacterial properties. Okay, that's one thing. There are nanomaterials that are being considered for use in construction materials. So we're talking about using something very small to optimize the performance of the largest structures that we see, that we live in. And definitely in medicine, there are just really tons of potential medical applications out there. One that I have really just read about and I've heard about in a seminar is potential cancer treatments. If you have cancer cells that are amongst healthy cells, right, and you want to just get rid of the cancer cells, and you don't want to harm or damage the healthy cells, right? Well, current ways of dealing with it are kind of brute force chemotherapy and radiation treatments. They really just affect everything, and they can't distinguish between healthy and not healthy. Those standard methods will cause sickness, yes, because they damage the healthy cells while they take out the bad cells. Okay, so what you would really like is to be able to just target, pinpoint the ones that are unhealthy, the cancerous cells, and kill those. Right? So one thing that I've seen are nanoparticles that have, we would say that they're functionalized. The surface has certain chemical groups on the outside that are complementary to chemical groups on the surface of the cancerous cells. It's like a lock and a key. It only fits those. All right, so you have the healthy cells that don't have those chemical groups, right? And so your nanoparticles can be brought in, can be sent to the general area, but then they will find the cancerous cells. Right? So that's one way you could detect the presence, before it really spreads. You could detect it. But then there's also the potential to have those nanoparticles loaded with a medication. Right? So you send in your particles, they find the cells, and then they deliver the medication. And this hasn't, as far as I know, hasn't been done in real living systems. It's been done in a dish in a lab, but it's going to happen. It's really, really incredible. Say you have a pile of nuts and bolts on the table. All right, and you say, well, I need to separate the nuts from the bolts. They're very tiny, though. I need to separate them. And for some reason, you're wearing big snow mittens, right? So, yeah, try and get in there and separate the nuts and the bolts when your hands are covered in these huge mittens. You can't do it. You're just going to knock everything out, right? Think of that as like your standard treatment, your brute force treatment of a cancer. So instead, take off the gloves. You have your fingers. Now you can get in there and you can pick up, you can separate, you can find the things you want and take them out. So this particular application we're talking about here is akin to taking off the gloves to give you that precision that you need. My research is on conducting polymer nanomaterials. And you can think of it sort of as bringing together two different fields. So there's conducting polymers and there's nanoscience. Okay? Conducting polymers are kind of plastic. They happen to conduct electricity. And that's unusual. If you think about most of the plastics, the conventional polymers that we deal with, plastic water bottle, for example, they've got lots of useful properties. They're very versatile. They can be flexible. They can withstand changes in temperature. But a thing they can't do is conduct electricity. Okay, so there is something, about the chemical structure of the conducting polymers that allows them to do that. They have mobile charge carriers similar to metals. Not the same as metals, but similar. So they have some conductivity, and that conductivity can vary. So you've got conducting polymers. And what I've done, or what I am doing, is looking at them from the nanoscale perspective. Right? So I think of it sometimes as teaching an old dog some new tricks. Conducting polymers are pretty well known, and the one I specifically work with is called polyaniline, and it's been around forever. It's been under pretty intense study for the last several decades, but for a long time not as any kind of nano material. Nobody really thought of it that way. In the last couple of years, maybe in the last 5 years or so, 6 years, it's been discovered that it's really quite easy to make polyaniline or to prepare polyaniline as a nanomaterial, specifically as nanofibers. So fiber, think of it as something that's long but thin. So in this case, we're talking about fibers that are a few micrometers long and less than 100 nanometers in diameter. If we go back to the size of a human hair, right? So a human hair is a fiber also, right? It's very long, it's very thin. These polymer nanofibers are very long, very thin, but the approximate size of these nanofibers, you would have to line 1,000 of them up side by side, right? Like this, 1,000 of them up side by side to reach the length of a human hair. The thing that's interesting to me very much is the morphology, the size and the shape of these nanoparticles. Now, in this case, they're fibers, so they have a fibrous morphology. Some of them are smooth, some of them are rough. It depends on how they're prepared and what kind of treatment they're brought through. Each nanofiber is really made up of, it's really a bundle of smaller particles. All right? See, I haven't really defined that yet. The polymer itself, a polymer is just a long molecule. That's all it is. Okay? It's got repeating units, and it's just extremely long. The conducting polymer nanofibers are just bundles of those polymer strands. Okay, so what I'm trying to do, or maybe uncover, are some ways to modify the strands, change the chemical structure of the strands, so that I can controllably alter the morphology of the nanomaterial. I would say, truthfully, I'm mostly interested from just kind of a pure research perspective, basic science perspective. I'm interested in how chemical structure affects nanoscale morphology. But it is certainly possible that different kinds of morphologies will have different kinds of applications. So I would probably leave it at that. Something else that has kind of grown out of that recently, so conducting polymer nanomaterials is my main field of study, but there's a lot of interest in hybrid nanomaterials. What that means, what I mean by that is bringing in several kinds of nanoparticle and combining the useful properties of each of them. Okay, so two things that come together to make something better. So I'm working with the conducting polymers and combining them with metal nanoparticles, like gold nanoparticles. Right? And the metal particles are smaller than the polymer nanofibers. So how you can think of it is like a pretzel and salt. Okay, right? So you have like a long pretzel, and the salt is dotting the surface of the pretzel. All right? So these small metal particles decorating the surface of the polymer nanofibers. And what I'm trying to do is optimize the interaction of the metal with the polymer. If you go back to the pretzel again, all right? If you have, like, say you want to make some pretzels. You've got the pretzel dough, and you've got the pile of salt, and the dough is dry, and you just like shake some salt over it. It's not going to stick very well. You get some of it to stick. But if you wet the surface of the dough and then you sprinkle the salt on it, the salt particles are going to stick. Right? So I'm trying to find basically better ways to make the gold nanoparticles stick to the polymer surface, but rather than just making it wet, I'm working on modifying the chemical groups on the surfaces of both components so that they want to interact more strongly. So the reason that you would want to have gold nanoparticles with these conducting polymer nanofibers, well, there's two reasons. Gold is an excellent conductor, and in order for a signal to be read from the sensor, it's usually some kind of electrical signal. So the gold is conductive, the polymer is conductive. So they both kind of serve to get the signal to whatever your larger device is, like a computer that's taking the information and giving you a readout. But there's more than that. With this particular system, so if you've got the polymer nanofibers, you've got the gold nanoparticles, and then you've got the analyte, glucose, coming in to interact with the glucose oxidase, the enzyme. That interaction between glucose and glucose oxidase produces another chemical. It produces hydrogen peroxide. So what we're actually doing is measuring the amount of hydrogen peroxide that's produced. It's not so much directly the glucose, right? The glucose comes in, does a reaction with glucose oxidase, and the product of that is what we're measuring. And the way we measure that is, well, it reacts then with the gold nanoparticles. Okay, so it's glucose reacts with glucose oxidase, produces hydrogen peroxide. The hydrogen peroxide reacts with the gold, and there's a current produced by that. So it's several steps separating between the thing you are interested in and what ultimately gives you the signal that you're reading, the electrical signal. One of the advantages of nanomaterials compared to bulk materials is the huge increase in surface area. If you have a sensor, there's always some part of the sensor that's got to interact with its environment. The analyte molecules come in and interact with that chemical sensor, and that's where the work happens, at the interface between the analyte and the sensing material. Okay. Now, a lot of sensors are like just flat surfaces. Right? So imagine a sheet of paper. And the surface area that you have to work with is the top of the paper, the very thin edges of the paper, and maybe the bottom. Okay? So you say, all right, that's my surface area. But there's a lot of material that's not getting used. That's everything that's inside that sheet of paper. Now, that may seem, well, you may say it's a sheet of paper. What could possibly be inside? But remember, we're talking about molecules, right? These really, really small species. So there's plenty of space inside that sheet of paper. There's a lot of surface area that goes unused. So here's what you do. You tear off the corner of that sheet of paper. Right? Now you have exposed surface area that was previously inside the sheet. Right? So you tear off the corner. Now you have this new surface area and this new surface area. Right? Tore it apart. Keep doing that. Keep tearing it into smaller and smaller and smaller pieces. You keep exposing more surface area. Well, that's the same thing as going from a bulk material to a nanoscale material. You go from a limited amount of surface area for a given amount of sensing substance, for a given amount of material, bring it down to the nanoscale, and suddenly you've got a huge increase in surface area. All these particles with all this new surface area. So what that gives you is lots more space, lots more surface for your analyte to interact with. But you need to get as much of your analyte to interact with the sensor as possible to have useful sensitivity, to have like reasonable sensitivity. If all you have is the top surface, if all you have is a flat sheet and a top surface, there are a couple things that can happen. It could get loaded very quickly, like kind of saturated, and then you just don't get any more activity out of it. There may be useful material inside your surface, but the molecules have to diffuse through it, and that takes time. If you want a sensor, you want to be able to take your sensor and like dip it in a solution and have it work very quickly. So you don't want to have to wait for molecules to burrow through a surface and get to get all of the possible interaction out of it. You want to work with a high surface area material, nanoparticles with lots of surface area, so that detection can be quicker and sensitivity can be improved and limits of detection can be lowered, right? So you optimize your sensor. When you are trying to detect something, right? You've got a sensor and it's meant to detect glucose, for example. The thing you're interested in is the analyte. So in that case, glucose is the analyte. If you've got a sensor that's supposed to detect methane gas, methane is the analyte. Right? So whatever it is that you're looking for, we would consider that the analyte. The particular application that we were first thinking of, that a colleague and I were thinking of, was monitoring glucose levels in the blood. All right? So this would be like a sensor that a person with diabetes could use to measure or monitor their blood glucose, because diabetes it can result in really dangerously high blood glucose. Okay, so you want a way to quickly and easily measure those levels. Now, there's plenty of methods out there. There are commercially available devices. You can use them easily. But it doesn't mean that there's not room for improvement. In my case, what we're looking at is how easy it is to fabricate the sensing material. Okay, so the polymer nanofiber with the gold nanoparticles is actually 2/3 of a sensing material. The third part is an enzyme called glucose oxidase that directly interacts or reacts with glucose. So it's a 3-component system. But what we're interested in right now is, can we optimize the polymer and the metal nanoparticle part of it? There's work out there like this, all right? And this is what kind of brought us in. We said, oh, look, this is out there already. There's got to be, I bet you there's a better way to do it. And what we're trying to do is take the fabrication of the sensing material down from several steps to a single step. And so if it's easier to make, you can do it, more easily and perhaps more quickly. And we think ultimately the method we're working on is going to improve the sensitivity of the sensor, so a better sensor. If you wanted to use this sensing material in a device, it would probably be very much like what's already out there. Like a test strip, you would take some blood and just put the test strip in contact with the blood, and then it gets soaked into the sensing part of it, the strip of it, right? And then it measures the glucose, and you get a readout. So it's not so much the device that we're trying to improve, it's the actual chemical sensing portion of it. Yeah, we believe that by improving on the fabrication method of it, that we should be able to get better sensitivity. And also, glucose is just one kind of thing to try and detect. Because this system uses an enzyme, glucose oxidase, it's specific to glucose. Well, there's other enzymes out there, so there could be we could extend this to other enzymes and whatever it is that they are specific to. Okay, so I'm at Queensborough Community College. It's one of the 2-year schools in the CUNY system. We have undergraduates, right? We don't have graduate students. We don't have postdocs. We have heavy teaching loads. But we also are all research active in our department, in the chemistry department. So if you want to get stuff done, you got to get the best of who is around you. Right? And these are our undergraduates. We have some really sharp students that we've gotten involved. And what's I think is very important is that these are first and second year undergraduates. Right? These are people that have maybe taken General Chemistry 1, probably not even in organic chemistry yet. If you go to a 4-year school, those first and second year students are not getting involved in research, not being recruited into anybody's labs. They're usually considered underqualified. The students that we've got are basically my hands in the lab, right? The project is of my design, the projects are my design, and the students are there to work in the lab and conduct the experiments. Now, they may not necessarily be at the point where they're designing their own experiments and or interpreting the data from a broader perspective, but with time they begin to learn how to do this. It's really just a matter of experience, of some training. They get the confidence to work independently. They understand what to do with their data and how to talk about it, and so we can have conversations about what they're working on, and we can push the project in a new direction if it needs to go that way. And all of our students end up presenting their research. They all go to local professional meetings, regional professional meetings, national professional meetings, usually American Chemical Society meetings, plus some other professional societies. They do oral presentations, they do poster presentations with their peers from the 4-year schools. I think presenting is important for students for the same reason it's important for anybody. You need to be able to talk about your work. You need to feel ownership of your work. And that's what the students are coming away with when they do this. So maybe they work with me for a year, or if I'm lucky I get them a little longer, maybe a year and a half. And they've learned not only a bunch of lab techniques, they've not only gotten their hands on instruments that they would have to wait much longer to use at a 4-year school, but they've also come to the conclusion that this is their project, and they know the most about it. They know more than anybody else as far as what have they done. It's theirs. And so that really goes a long way, and of course it looks great on a resume when they're transferring, if they graduate or if they transfer to a 4-year college. So it's really a great thing for them to be able to have this experience with us and then to and then to move on. I guess you could say we're trying to get them hooked early. You know, science is supposed to be for everybody. We should be able to defy some stereotypes about who does science, you know? And so we're trying to do that. We're recruiting students right away. We're showing them that they can do this. We're giving them the first-hand experience. At Queensborough, we're very fortunate that we've got a lot of modern instrumentation. The National Science Foundation agrees that undergraduates should be brought into research, all right? So they're definitely willing to provide funding if you can justify it, right? So they have these major research instrumentation program, and I pulled together a grant proposal with some of my colleagues in chemistry and in geology so that we could get a scanning electron microscope. So a scanning electron microscope is usually sort of an instrument that requires its own room, requires a lot of maintenance and upkeep, requires like a dedicated operator, someone to keep this thing running. But there are now a couple models that are like benchtop versions. You can put them on this table. You can put them in your car if you need to and like, you know, set it up someplace else. So the NSF awarded us the grant. We have this scanning electron microscope, this tabletop version of the scanning electron microscope, and it's absolutely essential to the work that I'm doing. So this is our way of looking at the nanomaterials that I'm preparing and that also my colleagues in the department are preparing. It's part of a more collaborative research program. I have colleagues at the College of Staten Island that I work with because they have more advanced imaging facilities. So we do as much as we can at Queensborough. The students get their hands on the instruments there, they have their projects going, and then when it's time to bring it up to the next level, I take them over to Staten Island, and we kind of can finish the work on more sophisticated, higher resolution instrumentation. I think it's important to realize, for myself anyway, that I'm just in one corner of a very huge field. Right? So, okay, you could say nano is small, but, you know, the field is huge. I like to think of this as a set of tools, right? Nanoscience as a set of tools, nanotechnology as a set of tools. So, you know, I can build something using a conventional toolbox, you know, screwdriver, hammer, and pliers and stuff, and I can build something with my hands. And that we would consider a very, you know, like a bulk material or a bulk object. You have surgeons that can do minimally invasive surgery using very small, very specialized tools, right? And you can get into places, you know, inside the body without having to really, well, open up the body, right? New tools. What I see nanoscience and nanotechnology as being able to do is to give us a new toolbox to work with. Okay, so we're talking about smaller tools to build smaller machines, and that gets us back to the idea of being able to interact with the smallest, and that brings us back to being able to make extremely small particles that can interact with the world around us in, you know, very new and very different ways and very useful ways.
Mm-hmm.